Hi, my name is Jimena de Leon from theme number 2. Then, we will talk about the cell cycle. The cell cycle is the process through which a cell passes every time it is divided. The cell cycle consists of a series of steps that will be explained below.
I am Shirley Casiano and I will be talking about the first phase of the interphase. The first phase is called G1 or also known as the ground phase and is where the cell grows and doubles organelles. Right there it performs metabolic functions and accumulates material to replicate DNA. But what is DNA? It's a protein found in the nucleus of the cell and is made of two coil chains that form a double helix, and this molecule is large. It needs to be organized into more compact structures, and that is why it is coiled into proteins called histones. Then the DNA that is already formed with histone forms a new structure called nucleosomes, and this prevents DNA from getting tangled. The nucleosomes then come together and form fibers known as a chromatin. In this phase G1, chromatin could be observed as a ball of thread in the nucleus of the cell.
During the S phase, the cell replicates its DNA, that is in the form of chromatin. At the cell phase of division, the chromatin condenses and becomes visible as a chromosome. The chromosome, which have an X-like structure, consists for two individual chromatids which join in narrow place called centromer.
Hello, my name is Nestor Castillo and I will explain the G2 phase. The cell has grown, DNA has been replicated, and now the cell is almost ready to divide. This last stage is all about prepping the cell for mitosis or meiosis. During G2 phase, the cell has to grow some more and produce any molecules it still needs to divide. This includes assembly of the microtubulus, which will make up an important structure called the spring during mitosis. At this time, the cell also has to duplicate its organelles so that each daughter cell has everything it needs. At the end of G2 phase, there is another checkpoint. The cell needs to check to make sure that all the proper structures are there and ready for cell division. This is especially important for the microtubules and replicate DNA from the S phase.
Hi, my name is Fernanda Delgado and I'm going to talk about mitosis. Mitosis occurs in 4 phases. The first one is prophase. After the sister chromatids form, two structures called centrosomes move away from each other outside of the nucleus. As they move to opposite sides of the cell, the centrosomes form something called the mitotic spindle. The mitotic spindle will eventually be responsible for separating the identical sister chromatids into new cells and is made up of long protein strands called microtubules. The nuclear membrane breaks apart into a bunch of membrane vesicles. Once the nuclear envelope breaks apart, the sister chromatids that were stuck inside the nucleus break free. Now that the nucleus protective cover is gone, kinetochore microtubules move near the sister chromatids and attach to them at the centromer. Now, these kinetochore microtubules are anchored at opposite poles on either end of the cell, so they're extending themselves toward the sister chromatids and connecting them to one of the edges of the cell. The second one is metaphase. Since the microtubules are anchored at opposite ends of the cell, their back and forth pulling on different sides of the sister chromatids gradually shifts the sister chromatids to the middle of the cell. This equal and opposite tension causes the sister chromatids to align along an imaginary line trailing down the middle of the cell. Now, the sister chromatids must be equitably distributed across the mitotic spindle. The third one, anaphase. During anaphase, the centromers at the center of the sister chromatids are severed. Anaphase ensures that each chromosome receives identical copies of the parent cell's DNA. The sister chromatids split apart down the middle at their centromere and become individual identical chromosomes. And the last one is telophase. The mitotic spindle is disassembled. The nuclear membrane is restructured around each group chromosomes, so that the cell now has two nuclei with the same amount of genetic information. Additionally, chromosomes begin to divide and nuclei begin to form. Next is cytokinesis.
Hi, my name is Kelly Giovanna Celestino Castillo and I'm going talking about the cytokinesis. Once mitosis has concluded, cytoplasm is divided by a process known as cytokinesis, in which the cytoplasmic membrane begins to invaginate and together with the ring composed of actin and meiosis proteins, press the center of the cell until it separates into two. Each daughter cell receives a nucleus and part of the cytoplasmic of the stem cell. Each one begins covered by its own plasma membrane. But in the cell plant is different because it has a rigid cell wall. Its division by contraction is not possible. To separate the cytoplasm, a series of carbohydrate phyllid vesicles from the Golgi apparatus will migrate and be sorted along to equator between both nuclei. Subsequently, the vesicles will fuse and form a cell plate. The cell plate will grow until it reaches the plasma membrane to fuse with it, dividing the cytoplasmic. Finally, the two daughter cells will have their own plasma membrane and cell wall.